Okay, so this is a video on the extravasation of leukocytes. So I'm going to talk about three different leukocytes: neutrophils, inflammatory monocytes, and naive lymphocytes. Before I talk about that and the different molecules that are involved in recruiting these molecules or these cells to a site of damage. So this is a knife penetrating some tissue here. So obviously on the surface of your skin there would be bacteria, and the bacteria would get inside of your cells. So you need some sort of phagocytic and inflammatory response, and then which leads to an immune response, an innate immune response, and then an adaptive immune response after that. So I'm going to talk about each one of these, but first I just want to briefly touch on what the different processes that I'm going to actually talk about here mean. So when I say that this molecule is involved in rolling, what that means is that this cell is literally just rolling along this blood vessel. So this is a capillary right here, these two red boundaries, and this cell is just rolling along. So he's just rolling, hanging out. It would be a little bit bigger, it would be blocking the whole vessel like this, but just for illustrative purposes, it's rolling along. Then you have what is called starts to it is bound to some sort of molecule that activates it. After that, so I'll just write this down: rolling, activation, and then here you have adhesion. Okay? After adhesion, you have what's called transendothelial migration, right? So it's leaving the vein or the capillary and going into the tissue. So think about something like a monocyte. In the blood, it is a monocyte, and then in the tissue, it's a macrophage. Remember that. So we're going to be talking about those in a second. So now I'm going to talk about the three different types of cells and what they're involved in. So there's three classes of molecules that are here. So for the rolling portion, you have the selectins. For the activation, arrest, or adhesion here, and the transendothelial migration, you have integrins and IgG or Ig superfamily members. And then for pretty much the entire time, you have chemokines that are involved in this. So I'm going to talk about each one of those here. So first to arrive at the site of an infection is a neutrophil. I already did a video on what neutrophils are, so I encourage you to check that out, or any other video online about it, just to understand what they are and what they do. So the molecules that are involved in rolling for neutrophils are L-selectin, PSGL1. Now, once they're done rolling and they need to be activated, the ones that are going to be involved in the activation of them are interleukin 8, IL-8, and what's called macrophage inflammatory protein 1b, or MIP1b. So I'll write it as MIP1b, and this is also known as CCL4, and we'll talk about that briefly in a second. Next, the molecules involved in adhesion for neutrophils are LFA1 and MAC1. So what neutrophils do is they're the first to the site of inflammation. So you have this inflammation happening here, obviously, because of this knife, you're bleeding, and there's bacteria coming in, so it responds to C5a, which is part of complement. It responds to bacterial peptides that contain N-formal peptides, and then it also responds to leukotrienes. Remember that leukotrienes is kind of like histamine, and they activate things like cis-LT and 5-lipoxygenase, which are involved in the inflammatory response. Next, we're going to talk about the monocytes. So I'll just label it as a monocyte, even though in the tissue it's technically a macrophage. So inflammatory monocytes. Here, the molecule involved in rolling is L-selectin. The molecules involved in activation are actually just one: monocyte chemoattractant protein 1, or MCP1, much like the MIP1, also known as CCL2. CCL2. In adhesion, they have VLA4. VLA4. That's a bad 4. VLA4. So what monocytes do is they come after, so they're the second ones to show up. So they're second to show up, and they respond to bacterial peptide fragments and complement fragments within this inflamed tissue here. Last, we have naive lymphocytes, right? So like B cells and T cells, when they are naive before they become mature lymphocytes. So naive lymphocytes here. Enrolling, this is going to get a little busy here. Enrolling, they have L-selectin, LFA1, which is similar to the adhesion molecule used for neutrophils, and then VLA4, which is similar for the adhesion molecule used in monocytes, VLA4. And these are in their low affinity forms. For activation, there are a variety, so I'll list them here. CCL21, CCL19, CXL, CXCL 12, and CXCL 13, I believe it is. Let me just check. Yeah, 13. So 12 is for T cells, and 13 is for B cells. Adhesion, they use the same molecules as before, VLA and LFA1. Okay, so what they do is they travel across the high endothelial venules to enter into the lymph node. And I made a video on what the lymph node was and what its function was. So that's it for the molecules that are involved in the extravasation of leukocytes.